Good morning children, welcome to AIMS India's online classes. This is biology session. You are watching Anatomy of Flowering Plants part 3. So, we have successfully completed about the types of tissues in plants. Types of tissues in plants are divided into two, that is meristematic tissue and permanent tissue. Okay. In the meristematic tissue, we have discussed about the apical meristem, intercalary meristem and lateral meristems. Apical meristems help in growth in length, intercalary meristem help in elongation of the organs, and lateral meristems help in increase in the girth of the stem. Okay. And about permanent tissues, the tissues in which the cells last the power of cell division. Permanent tissues are again divided into two types. What are they, children? They are simple permanent tissue and complex permanent tissue. In the simple permanent tissue, we discussed about the parenchyma, collenchyma and sclerenchyma, isn't it? The complex permanent tissue, we have studied about the xylem and phloem. Okay? And finally, the tissues together form the tissue system, which are of three types in plants. What are they? Epidermal tissue system, ground tissue system and vascular tissue system. So, we have completed the epidermal tissue system. In the today's session, we are going to discuss about the ground tissue system. Okay. Are you ready, children? Let us proceed. The ground tissue system. What is ground tissue system? It consists of simple tissues and it is derived partly from periderm and partly from plerome. Okay. So, all the tissues except epidermis and vascular bundles constitute the ground tissue. It consists of simple tissues such as parenchyma, collenchyma and sclerenchyma. Okay, the parenchymatous cells are present in cortex, pericycle, pith and medullary rays. In leaves, the ground tissue consists of thin walled chloroplast containing cells called mesophyll tissue. Okay. This mesophyll tissue only divided into palisade parenchyma and spongy parenchyma. Okay. The internal organization of the leaf is such that it suits the process of photosynthesis. Okay. The parenchymatous cells present in the internal organs. Okay. If you take a cross section of the plant parts, we study these parts like cortex, pericycle, pith, medullary rays, etc. Okay. So, the vascular the ground tissue system. So, this is about the ground tissue system, the basic we are discussing. So, as I told you, it is derived partly from the periderm or peribellum and partly from plerome. Okay. It is of two types, extrastellar. Extrastellar it is called as cortex and intrastellar. Intrastellar include the pericycle and the pith and medullary rays. This is intrastellar part. Okay. So, in leaves it consists of chloroplast containing the cells and is called mesophyll tissue. Next, vascular tissue. The vascular tissue includes stele and the central column of the plant body that consists of vascular bundles and pith. Okay, the vascular tissue system consists of the complex tissues. What are they? Xylem and phloem. The xylem and phloem together constitute vascular bundles. A vascular bundle is made up of a xylem and a phloem. Each vascular bundle comprises of xylem and phloem and the cambium if present. Okay. So, you can see here the vascular tissue is in the middle. Okay. This is the pericycle part, this is the section of stem, epidermis. Okay. If it is root, root hairs are present. Okay. Endodermis, vascular cambium, primary phloem, primary xylem, the pericycle. These are the parts internally. The vascular tissue system consists of complex tissues. What are they? Xylem and phloem. Okay, the xylem and phloem together constitute the vascular bundle. So, you can see in dicot the presence of cambium between the xylem and phloem. If the cambium is present, such type of vascular bundles are called open type of vascular bundles. Open type of vascular bundles. The vascular bundles without cambium, they are called closed type of vascular bundles. Okay. And another type of vascular bundles, okay, one is open type of vascular bundles in which the cambium is present, closed type of vascular bundles in which cambium is absent between the xylem and phloem tissue, and the radial vascular bundles, xylem and phloem are arranged alternate manner on the different radii. Okay. And another type of vascular bundle, conjoint vascular bundles. Conjoint vascular bundles, these are in which the xylem and phloem are situated at the same radius of vascular bundle. Okay. So, vascular bundle is divided into different types, basically open and closed. Open means cambium is present, closed means cambium is absent. Next, radial. Radial means the xylem and phloem on different radii. Conjoint. Conjoint means xylem and phloem on same radii. Again, conjoint vascular bundles are divided into two types, collateral and bicollateral. Collateral means the phloem on outer side and xylem on inner side. So, you can see here in dicot and monocot stems how the arrangement of vascular bundles. The vascular bundles are scattered in monocot stems, whereas the vascular bundles are arranged in a systematic manner in the dicot stem. Okay. So, in the monocot stem, in the ground tissue itself, the vascular bundles are scattered. So, by observing this under the microscope, you can easily identify whether the plant is monocot or dicot. Okay. So, what is bicollateral vascular bundle means? Xylem is sandwiched between the outer and inner phloem. Okay. So, another type of vascular bundle, concentric vascular bundles. Concentric vascular bundles means one surrounds the other. Okay. Xylem surrounds the phloem. So, these concentric vascular bundles are again divided into amphicribal or hadrocentric and amphivasal or leptocentric. This you will study in higher classes. Amphicribal means phloem surrounds xylem. Amphivasal means xylem surrounds the phloem. Okay. So, let us proceed. The dicotyledonous root first. Okay. Here the outermost layer is epidermis. The presence of unicellular root hairs in epidermis is the characteristic features. What are the characteristic features of dicotyledonous root? Means the dicotyledonous roots possess a outer layer epidermis, the presence of unicellular root hairs in epidermis. The cortex consists of many layers, thin walled parenchyma cells with intercellular spaces. The innermost layer of cortex is endodermis. The endodermis consists of single layered barrel shaped cells without intercellular spaces, endodermis. Okay, endodermis consists of single layered barrel shaped cells without intercellular spaces. And another characteristic, very important characteristic feature of this dicotyledonous roots is presence of Casparian strips in endodermis. Presence of Casparian strips in endodermis. So, next to the endodermis, there are few layers of parenchymatous cells which form the pericycle. Okay, the pericycle is present in the roots. The initiation of lateral root and vascular cambium during the secondary growth takes place from the cells of pericycle. It is the characteristic feature of dicotyledonous roots only. Secondary growth we can see. Okay. So, this is the dicotyledonous root. You can see this is the outer epidermis or epiblema. So, it possesses unicellular root hairs. Okay. So, beneath the epiblema, the layer of cells are present, it is the cortex. Okay. The cortex is the innermost layer. The innermost layer of the cortex is called the endodermis. This is the endodermis. Okay. It possesses the barrel shaped cells, and with without intercellular spaces. There is no intercellular spaces, see here. Okay. And the presence of Casparian strips, they are present in the dicotyledonous roots. And next to endodermis, there is a layer that is called as a pericycle. Okay. This pericycle is the region from where the lateral roots and vascular cambium develops during the secondary growth. It also possesses the pith. This is protoxylem, okay, towards periphery, and metaxylem towards the center. Okay. Protoxylem means first formed xylem, metaxylem means the matured xylem. Okay. And the phloem tissue, see this is scattered. The barrel shaped cells you can see in the endodermis. These are the barrel shaped cells. Next, the parenchymatous cells present in between the xylem and phloem is called conjunctive tissue. Okay. The parenchymatous cells present in between the xylem and phloem, it is called as conjunctive tissue. The number of xylem and phloem bundles is 3 to 4. Okay. Previously we saw 4, protoxylem, metaxylem, the layers. Okay. All the tissues on the inner side of the endodermis, such as pericycle, vascular bundles and pith, constitute the stele. Next, monocot root or monocotyledonous root. The monocotyledonous root have similar tissues as in the dicot, except it contains more than 6 xylem bundles called polyarch xylem. Okay. So, you can see polyarch xylem, more the xylem bundles are present. So, similar root hairs are present, epiblema, then beneath that cortex and next to cortex endodermis, and then phloem, pith, protoxylem, metaxylem. These are the parts of a monocot root section. It contains more than 6 xylem bundles. Okay, more than 6. So, you can see 7 here. The pith is large and well developed. The pith is large and well developed in monocots. Do not undergo any secondary growth because pericycle is absent. Okay. So, there is no secondary growth in monocot roots. Next, dicotyledonous stem. So, before going to dicotyledonous stem, let us have a brief discussion difference between the dicot root and monocot root. In dicot root, the cortex is comparatively narrow, whereas in monocot it is very wide. If you compare the endodermis, in dicot root, less thickened and Casparian strips are more prominent, whereas in monocot roots, the endodermis, if you consider, it later becomes highly thickened. Casparian strips are visible only in young roots. Okay. Then in dicot roots, passage cells if you take, generally absent. In monocot root, generally the passage cells occur opposite to the protoxylem point, and the pericycle in dicot produces the lateral roots, cork cambium and part of vascular cambium, whereas in monocots, the pericycle produces only lateral roots only, it is not visible. Whereas in dicot root, the vascular bundles if you take 2 to 5 or sometimes 8, but whereas in monocot 8 or more in number vascular bundles. And if you take the pith in dicot root either absent or very small, but whereas in monocot root the pith is very large and well developed. Now come to dicot stem. The outermost layer is epidermis in the dicot stem. The epidermis covered with a thin layer of cuticle and has trichomes and few stomata. The cells arranged in multiple layers in between the epidermis and pericycle, it constitutes the cortex. The cortex has 3 subzones. What are they? Hypodermis, cortical layer and endodermis. Hypodermis, the cortex divided into 3 layers. The first one is hypodermis. The hypodermis, a few layers of collenchymatous cells below the epidermis are present. Then cortical layers consist of rounded thin walled parenchymatous cells with intercellular spaces. The endodermis, if you take, it is the innermost layer of cortex, and the cells are rich in starch grains and are referred to as starch sheath. Okay, see, this is the structure of Dicotyledon stem. So, epidermal hairs are present. Epidermis is the outermost layer. Next to epidermis, hypodermis. This hypodermis is divided into cortex, cortical layer, and so this cortex has 3 subzones, that is hypodermis, then cortical cells or cortical layer, then endodermis. The endodermis is the innermost layer of the cortex. It has cells rich in starch grains and referred to as starch sheath. It is also called a starch sheath, this endodermis. Okay. Then the vascular bundles, they are present to periphery, okay, facing towards the center, and the pith is small. Okay. So, the pericycle, if you take, it is present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. The medullary rays are present. A few layers of the radially placed parenchymatous cells present in between the vascular bundles, they are called medullary rays. Okay. A large number of vascular bundles are arranged in the form of a ring. Okay. Each vascular bundle is conjoined. What is conjoined? Xylem and phloem on same radii. Conjoined, open type. Open means they possess cambium. Okay. And endark protoxylem. Endark protoxylem is present. Okay. The central portion of the stem constitutes the pith. This is the pith. Okay. This is a dicotyledonous stem. Now come to monocotyledonous stem. It has similar tissues with the dicot stem except in following, that is sclerenchymatous hypodermis. The vascular bundles are scattered in the ground tissue and each vascular bundle is covered by bundle sheath cells. The vascular bundles are conjoined. Conjoined means xylem and phloem on the same radii, but closed because the cambium is absent. The peripheral vascular bundles are smaller than central one because vascular bundles are scattered. The phloem parenchyma is absent. The water containing cavities are present within the vascular bundles. Okay. So, this is no epidermal hairs here, see, in the monocot, only epidermis, hypodermis, the ground tissue, vascular bundles. Vascular bundles are scattered in the ground tissue. Okay. And okay, before going to end the class, let us discuss some differences between the monocot stem and dicot stem. If you consider the vascular bundles, in monocot, vascular bundles in ring, whereas in dicot, they are the vascular bundles in ring, whereas in monocot stem, they are scattered. The vascular bundles are conjoined, collateral or bicollateral and open type in dicot stem. The vascular bundles are conjoined, collateral and closed type in monocot stem. The bundle sheath is absent in dicot stem. Bundle sheath usually present in the monocot stem. The phloem parenchyma is present in the dicot stem. Phloem parenchyma is usually absent in the monocot stem. If you take pith or medulla, the made up of parenchymatous cells situated in the center of the stem, whereas pith is absent in the monocot stem. The ground tissue, if you take in dicot stem, differentiated into cortex and pith, whereas in monocot stem ground tissue is not differentiated into cortex and pith. You won't find here cortex or pith. It is a monocot stem. Okay, hypodermis. Hypodermis is cholenchymatous in dicot, whereas sclerenchymatous in. So this is the characteristic feature of monocot, sclerenchymatous hypodermis. Endodermis, if you take one layered starchy sheath which is usually not well differentiated, whereas in monocot the endodermis is absent. Here no endodermis, you can see. The next, pericycle. Pericycle made up of one or more layers of parenchymatous and or sclerenchymatous cells, but pericycle is absent in the monocot. Here in monocot stem there is no pericycle. Medullary rays, they are found in between the vascular bundles in dicot, but here medullary rays are absent in monocot. The secondary growth, if you see, in dicots there is a secondary growth, but here because pericycle is present, okay, vascular cambium, and here the pericycle is absent. There is no secondary growth in the monocot stem. Okay children, I hope you understood. Please read the book. Thank you for watching our video. Please subscribe our channel and press the bell icon for receiving latest updates.